Hi everyone, this is lecture 12, Mendel and the Gene Idea. So this is what you'll hopefully be able to do by the end of the lecture, and so I'm going to keep going for now. So, how are traits passed on or transmitted from parents to offspring? So that's basically what this whole lecture is going to cover. So each parent cell has two alleles for each character or trait. And so this is true for plants as well as humans. I think sometimes we can be human-centric, but what we know about genetics we learned from these plants. And so we can take that and apply it to humans towards the end of the lecture. But we've learned how you can separate chromosomes during meiosis, right? And then during fertilization, gametes come together to form a new organism or a zygote. And so that is how offspring end up getting one copy of each thing or each recipe for something. And then offspring have the full number of chromosomes. And so even though it seems very different, meiosis, and then we jump into genetics and it seems like it's a totally different thing, they are connected. So I hope it comes a little bit more easy as we go through. So everything we know about genetics really started with this guy, Gregor Mendel. So he's often called like the father of genetics. So Gregor Mendel was an Austrian monk, and he had a lot of time. He also really liked gardening, and so he ended up experimenting with different pea plants. So the like green peas that we eat, they do have like a full plant and flowers. And so he ended up experimenting with pea plants. And by doing so and having just kind of curiosity, he ended up discovering the basic principles of heredity by breeding these garden peas in very carefully planned experiments. And we learned a lot. So Mendel's approach to the study of heredity, or how things are passed on, allowed him to deduce principles that had remained elusive to others, meaning like he had a lot of patience and he was able to figure out things that a lot of people couldn't figure out yet. There were a lot of different hypotheses about inheritance and what gets passed on, and everybody thought it was just you just mash everything together basically, and that's what gets carried on in offspring. So yeah, in the 1800s, everybody had different ideas, but his work allowed quantitative data to show us like what these patterns are and how things do get passed on. So throughout this lecture, we're going to use the term character. And so the textbook, defines this as a heritable feature that varies among individuals, such as flower color. So something that can be inherited, like flower color, can be passed on, right? Hair color, hair texture, we can call that a character. So something that varies among individuals but can be passed on and inherited. And then different variants for a character, like either purple flower color or white flower color, these different variants are called, or each variant is called a trait. So a character is like the flower color, and then the different variants are traits. So Mendel, in a way, got really lucky. If he had used any other kind of plant, it could have showed him different patterns. But he got really lucky using these garden peas, and there were many advantages. So for him, they were available to him in many different varieties, and so that's kind of why he used them. But there were a lot of other advantages too, other than there being like a lot of varieties to work with. Peas reproduced quickly. They have many offspring, so each one of these peas in a pod is an offspring that you can count. And so in one generation, you can count 5 different, offspring. So, another feature that's really interesting is how, yeah, the matings can be controlled. So, if we look at this, the flower of these garden peas, so these smaller little kind of stick-looking things are called stamens. And then that central thing, longer, like thick kind of stem thing, is called a carpel. So that's called shown here. And so the flowers of the pea plants typically have both of those structures. And so they can self-pollinate. And then they can also cross-pollinate if the pollen from the stamens gets transferred onto the carpel of another flower. And so in this way, he was able to actually control the mating. So if on one plant you cut off the stamens and then just leave the carpel, and then you physically transfer the pollen, which is the gametes, from another plant onto the carpel, then you're controlling this mating. And then as you let this plant develop and produce seeds, and then those seeds are going to get planted and produce their own individuals, then you have a whole another generation. And so he chose to track a lot of different traits. At first, he only tracked one thing at a time, or one character at a time. And so he would track things that only had two distinct forms. So if this character is flower color, and there was only two forms of this trait of purple flower color or white flower color, he studied things that only had two different versions. Or if the plant was tall versus short, these kinds of plants, you do see that big kind of range of sizes, or not a range, I guess, just forms. And so he started with varieties that were true breeding. And so what that means is, like, if you let this flower self-pollinate, it will only ever have purple flowers. So yeah, if we go back here. So this purple flower plant is, if you let it self-pollinate, it'll only ever have purple flowers. And so that's what's called true breeding. It only has more of like the same exact kind. And then same for this white flower. If he had let them self-pollinate, then it would only ever give white flowers. And so that's what, throughout these lectures, if something says true breeding, it just means, like, if you let it self-pollinate, it'll only ever have the same kind. So plants that produce offspring of the same variety when they self-pollinate. So in a typical experiment, what Mendel did was he, cross-mated two different, so contrasting true breeding varieties, like the purple and white flower plants, and he physically crossed them. And so by doing this, he kind of did this process called hybridization. Like, he made a hybrid of these two different varieties. So that's called hybridization, and what you make is a hybrid. And so he did a lot of these different crosses and matings of plants. And so he kind of needed a naming system, a naming scheme. And so those original true breeding parent plants are called the P generation, P for parent. And then what you cross, the next generation, is called the F1 generation. So those hybrid offspring are called the F1 generation. And F actually stands for filial, which means like son or daughter in Latin. So yeah, these are the sons or daughters of that first parent generation. And then after that, you can just name the different successive generations, F1, F2, F3, F4. And so there you have like a nice naming system for all these crosses. So the true breeding parent plants, if you were to just let them self-pollinate and they only ever have purple flowers, and then this one only ever had white flowers, and then you cross them, the original ones are the P generation, the offspring are the F1s. And you can either let those F1s self-pollinate, or you can cross-pollinate them as well to each other, or other things. And so then the next generation is the F2 generation. And so here's a figure of that. So if in the P generation, he crossed purple flowers with white flowers, in the F1 generation of hybrids, he saw that all the plants had purple flowers, and he was like, oh, that's interesting. It's almost like the white like disappeared, like where did that go? So when he crossed those contrasting or different true breeding white and purple flower pea plants, he found at first, okay, all the F1 hybrids are purple. Interesting. And so just out of curiosity, he was like, well, what happens if I let these self-pollinate or cross-pollinate? Let's just see what happens. Let's see if they also become out purple, or maybe the white comes back. Like, let's just see what happens. And so it turns out that the white did come back, the white flower color. So if you were to cross the F1s with each other or let them self-pollinate, he found a ratio, like every 3 purple ones, there's also 1 white one. And this ratio was a big pattern that he noticed. He didn't just cross one plant with another and then saw this. Like, he did this hundreds of times and counted up hundreds of flowers. And so you can actually see the data here. So for a total of 705 purple flowered plants, there was also 224 white flowered plants. And so in all these different generations, if you break down the ratio, it's like consistent with all these different like matings. There's this ratio of about 3 purple ones to 1 flower in the next generation. And so he was like, oh, that's super interesting. That has to mean something, right? That can't just be chance if like I'm seeing this ratio of 3 to 1 in every single batch that I mate. And if I add up and like do the ratio of all of this, it comes out 3 to 1. And so, yeah, he was like, this is really interesting. And so, considering the P generation and the F1 generation and the F2 generation, he, had time to think because he's just a monk. And so he had time to think. And so he reasoned that only the purple flower factor was affecting flower color in the F1s. Like the white was still there, but at that point in that generation, only the purple factor, because he didn't know what else to call it at that time, only the purple flower factor was affecting flower color in that generation, but then it's different in the next one. So he called the purple flower color a dominant trait and the white flower color a recessive trait. And so, yeah, that factor for white flowers, whatever like factors into these flowers being white, it wasn't diluted, it wasn't destroyed, it just was kind of hidden for a generation. And so this, was a huge deal, So the factor for white flower colors was not like diluted because it was white. It wasn't like a very diluted purple that came out white, and it wasn't destroyed. It's still there. It was just not being like allowed to be expressed because the other one was dominant. And so he gave these things these names. So he thought, that's really interesting. Well, let's study other stuff and let's see if I could see this pattern again. He was a good scientist for being a monk, So he reproduced experiments and he tried it with other things too. And so he saw this same pattern. He saw the same pattern of inheritance in 6 other pea plant characters. And so the character of like flower color, seed shape, pod color, pod shape, flower position, where's the flower on the plant, and then stem length. So he saw this pattern with each of these traits, or each of these characters, and that each had 2 different traits, so 2 different forms. Purple versus white flower color, yellow versus green seed color. So with all of these things that he was studying, he saw these patterns. He saw the pattern of roughly 3 to 1. And if you're like, how did he get that? So you would divide the bigger number, the 705, by the smaller number. And then so that gives you the 1. And then the other number is the ratio of roughly 3. So if you do the math, it's like 3.15, but roughly 3. And so he did this again and again and again. And look at all this data. It's so interesting. So for seed color, he looked at yellow versus green seeds. And so he found 6,000 yellow seeds and 2,000 green seeds. Again, that's like pretty even numbers. So that doesn't seem random. And so when he did the division again, that comes out to about 3 to 1. And so in this huge number of the totals, and in each generation, he saw this pattern. And the same for seed shape, roughly 3 to 1, pod color, pod shape, flower position, stem length. There's like a little bit of variation, but it's roughly 3 to 1. And so he's like, that has to be something. And so what he called heritable factors, now we know that they're genes, and we can call them genes. It's genes that get passed on from parents to offspring that like help code for these differences. But yeah, just by making all these observations and doing carefully planned experiments, he like really fell onto something, you know? So he developed a model to try to explain this 3 to 1 inheritance pattern that he observed in the F2 offspring. And so there are 4 related concepts that make up this model. And these concepts can be related to what we now know about genes and chromosomes. So this lecture is going to tie into the next few lectures because like we're going to track the genetics and stuff. But then it's also because it's chromosomes that we know that get passed on in meiosis, and then fertilization, and that's how all these things get passed on. So conclusion 1, he concluded through all this data, alternative versions of genes account for variations in inherited characters. So in like a population of one kind of organism, there's variation, you know? I guess off the top of my head, I'm thinking pigeons. You know, there are pigeons that are more solid in color. There are some that have like different colors, whatever. And so he reasoned that the variations come from alternative versions of genes, and those get passed along. So in a more simple example, the gene for flower color in pea plants, there are 2 different versions, one for purple flowers and one for white flowers, and that is what leads to these variations visually, because there's 2 different factors. And so now we call these alternative versions of a gene alleles. So for one gene, flower color, we can call the 2 different versions that they're 2 different alleles. There's an allele for purple flowers and there's an allele for white flowers. And each gene can be found on a specific location on a specific chromosome. So we have, you know, a bunch of different chromosomes, and then plants have a bunch of different chromosomes, but certain genes we know will always be located on this particular chromosome. So each gene that codes for something to be expressed, like the flower color, either purple or white, you know, now know that each gene lives or resides at a specific locus or like location, basically, on a specific chromosome. So that was his first conclusion. There's different versions of genes that account for variations in like purple flower color or white flower color. Okay, yeah, there's different versions of this thing that can be inherited. And so this is what I mean about the chromosomes. So here is one chromosome in a pair of homologous chromosomes. Remember, we get one set from, our mom, one set from our dad, and so we have a pair of homologous chromosomes. And so similarly, here for these plants. So they have a pair of homologous chromosomes, and so say this is like chromosome 1 in this plant. And so the gene for flower color is at the same location or locus, we call it when it's a location on a chromosome, the same locus on this chromosome as well as the other one because they're homologous chromosomes. And so you can have two of the same version or the same allele, or you can have one of each. But so this version at this locus has the allele for purple flowers. And so that's how this ends up purple. We're going to talk way more about gene expression in a future lecture, but so the gene is like a recipe for something. And so the recipe for the purple flowers has this small variation, CTAAT, CGGT. So this is the recipe to make the enzyme that places the purple color in the flowers. And the other allele actually is like a small change, a small difference that ends up not letting the enzyme be made correctly. And so that DNA sequence and that recipe results in an absence of that enzyme. And that's why they're white flower colors, because there's no purple pigment put into the flower. So yeah, that kind of shows you how alleles are different versions of one gene and how they can be different. Like there's literally a small difference in the DNA sequence that leads to these physical differences. So yeah, we'll talk a lot more about that in future lectures. But there are different versions of genes that can be inherited because they're on chromosomes. So conclusion 2, for each character, so again, like flower color, an organism inherits 2 alleles, one from each parent. And again, now we know that because we know meiosis and fertilization and how that gets passed on. And yeah, it's just super interesting. He was able to make this deduction without even knowing about chromosomes. He was just like, okay, something gets passed on and you must get one from each parent. And that results in 2 alleles. So the 2 alleles at a particular locus may be identical, like the true breeding plants of Mendel's pea generation. So again, true breeding means like if you let it self-pollinate, it'll only ever keep giving purple flowers. So the 2 alleles that those purple flowers have, they're both the purple flower color. And then the same for the white ones. It's true breeding, only ever makes white flowers, and so it must have both copies of the white flower color. So the 2 alleles can be identical, or they can be different, like the F1 hybrids. And so if I go back to this picture, these 2 alleles can exist in the same flower, but because one is dominant, the purple flower color is going to be expressed and the other one isn't. So that's why the ones in the F1 generation all look purple, despite the fact that they have both alleles. So conclusion 3, if the 2 alleles at a locus, again, or location, are different, then one of them, the dominant allele, determines the organism's appearance. So kind of how I was just saying, there can be a purple and a white allele, but if one is dominant, that's what's going to determine the appearance, like the purple flower color. The other allele, the recessive allele, has no noticeable effect on appearance. So the white allele, flower color, would be recessive. And so, yeah, in the flower color example, F1 plants had purple flowers because the allele for purple flowers is dominant and the white flower is recessive. You could kind of think about it. I like to say it's like the dominant one is a bully So whatever the dominant allele is, it just like bullies the recessive one into like submission, like I'm not even going to be expressed. And then the dominant one is the one that gets to be expressed because it bullies the other one basically. So the dominant one will be expressed, the other one kind of just hides until it gets a chance to be expressed. And so his fourth conclusion is something he called the law of segregation. So he came up with two laws of things. And so the law of segregation says that those two alleles for a heritable character separate or segregate during gamete formation and end up in different gametes. So again, now we know that that's like how meiosis works. So the chromosomes in meiosis get divided. And so those two alleles that exist on these chromosomes for a heritable character separate during gamete formation and end up in different gametes. And so, yeah, you have 4 different gametes, and so all the alleles get separated from each other. And so, thus, an egg or a sperm gets only one of the 2 alleles that are present in the organism. And so, again, now we know it's because of chromosomes. This segregation of alleles corresponds to that distribution of homologous chromosomes to the different gametes in meiosis. And so, what he came up with, this model, is Punnett squares. So the model accounts for that 3-to-1 ratio that was observed in all those different F2 generations. When he worked it out this way, it was like, oh, okay, yeah, no wonder this was like the pattern, 3-to-1 every time, or close to 3-to-1. So possible combinations of sperm and egg can be shown using what's called a Punnett square. You might or might not remember them from like high school. I think they're kind of fun, and hopefully you think that they're fun. So when you make a Punnett square, you're basically mapping out on the tops and sides possible gametes, so possible sperm or eggs. And then within the square, you're kind of mapping out potential fertilization and zygotes. So I'll show on the next slide, but yeah, you're kind of modeling out, okay, if this zygote or this gamete and this gamete fertilized, then it would lead to this combination. If this one and this one met, it would come to this combination. And so we can also help track these different traits by assigning letters to them. So for one character and the 2 different traits, we will give a letter. So like the letter P for purple flower color. And so we will say, if the purple flower color is dominant, we'll give it the capital letter, so capital P. And if the lowercase, then the lowercase letter would represent the recessive allele. So for one character, flower color, there's 2 different traits, purple or white. So then we would give the big P for purple and then a little p for white. And that is what we use to help track us through, different generations and through a Punnett square. So in this example, now we're like going back to the purple flowers and white flowers in the P generation. Now we know, okay, this purple flower was true breeding, so it's only ever going to give rise to purple flowers. And so we know that they have 2 copies of the dominant allele. So we would give this a big P and a big P. This white flower only ever has white flowers, and because there's no big P that's dominant and bullies it, like this is how the recessive one gets to be expressed if there's 2 copies of the recessive trait. So a little p and a little p, because it must have both copies to be a white flower. And so if we were thinking about meiosis and what possible gametes these could make, so imagine this is like a chromosome, and then this other one is another chromosome. If you divided them up, the gametes, there would actually be technically 2 big P's and then 2 little p's. So if you divided up these gametes, sorry, 4 of each actually. So if you divided up these gametes, there would be 4 gametes, and they would all have little p. And technically here, there would be 4 gametes, and they would all have big P. And so you're thinking about, yeah, the potential gametes they can have. And then when you cross them, you're thinking about, okay, it gets this copy from this parent and this copy from this parent. And so now this F1 offspring, even though it looks purple, it has 1 copy of each allele. The big P dominant for purple flowers, and it also has the little p recessive for white flowers. And so even though it looks purple, we know that it has a big P and a little p. Yeah, like the alleles for purple and the alleles for white. And so again, if we either let these self-fertilize or cross-fertilize with another thing, then it could have 2 different outcomes actually. So some of the gametes are going to have big P, and then some of the gametes are going to have little p. So you're kind of dividing them up like that. Like if you're imagining this is meiosis, there's actually, yeah, 2 big P and then 2 little p. And then, so if you cross these guys with each other or let them self-fertilize, then this is how it maps out in the Punnett square. So you're taking one plant that is F1, and so it has a big P, little p. And so you're planning out the gametes. So again, it could come out this or this. And so you would put 1 of each above these columns. And then you're crossing this plant with another plant of the same one. And so that's what you would put on the side. You're mapping out, okay, if it has a big P and a little p also, the gametes that it could make could either just have big P or just have little p. And then when they come together in fertilization, these 2 gametes, then this zygote, this flower gets made. And so this one will have big P, little p, because you did this and this. If you combine these 2, then you would have 2 big P's. If you did these 2, it would have a big P, little p, and then lastly, you would have one that is little p, little p. And so that is the ratio, right? Like 3 purple flowers to 1 flower. So if this was 1 generation, this is kind of like the odds that it would be. And so, yeah, this accounts for that ratio that he saw with all these different traits. And so Punnett squares, maybe like they're challenging at the beginning, but I hope they end up being fun for you. Maybe I'm a nerd.